Alright, so chapter 20, we're going to cover scientific names, kind of ranking, phylogenetic trees, homologies, nologies, clades, derived versus ancestral characters. We'll talk a little bit about molecular clock and domains of life and understanding the tree of life. We're actually going to go in a little bit of a reverse order here because I think it makes more sense to talk about kind of the different kinds of living things we have and then talk about how we classify them. So taxonomy is the science of naming and describing living and extinct organisms and also viruses. It's really a hierarchical system involving successive levels. Each group at any level is called a taxon. And early taxonomists thought things were either a plant or an animal, and that's it. You're a plant or you're an animal, nothing else. Later they realized that wasn't quite enough to categorize everything. So they recognized 5 kingdoms, Monera, which is all prokaryotic organisms, Protista, Plantae, fungi, and Animalia. We are now operating under a 3-domain system, which is supported by data from many sequenced genomes. It highlights the importance of single-celled organisms because there's actually two prokaryotic domains, bacteria and archaea, and then in eukarya, there's also many single-celled organisms. So under this system, all life belongs to one of 3 domains, bacteria, archaea, or eukarya. So if we look at taxonomy, the domain level is the most inclusive level, and the species level is the least inclusive level. So as we go from domain to species, the number of differences visible is actually going to decrease. Members in the same species are the most similar. Obviously, like if we were in a room with lots of people, I would say, look around, we all look different, right? Because we all do look different, but we are all the same species. So members of the same species have the most similarities with each other, but there are differences there as well. So this is one of those things that you'll need to memorize. So it goes domain, supergroup, kingdom, phylum, class, order, family, genus, species. If you've learned this before and weren't aware of supergroup is kind of a newer addition. So if we look at this, and let's say we look at the wolf, which is Canis lupus. So species Canis lupus, and then it's in the genus Canis, and then family Canidae, and then Carnivora, then Mammalia, then Chordata, then Animalia, and then Uniconta is the supergroup, and then it's in the domain Eukarya. I don't expect you to memorize all of this, but if we look back down to the species level, only the wolf is in that species. Then there's other members in the genus, and even more in the family, and even more in the order, and kind of so on and so forth there. So if we look at the domain level, again, the domain is the most inclusive, or there's the most variety of organisms that fall into a domain. Bacteria are prokaryotic organisms. They're unicellular. If they have a cell wall, it's composed of peptidoglycan. We've talked about bacteria multiple times before, so that should not be anything new to you. Archaea are prokaryotic and unicellular organisms. They do not have peptidoglycan in their cell wall. Typically, if they have cell walls, they're going to be made of protein. These guys can live in extreme environments like high temperatures or acidic pH. They don't only live in extreme environments, but that's what they're famous for. So for our class, we're going to kind of focus on that. Then we've got the domain Eukarya, which is all eukaryotic organisms. So everything with a nucleus falls into this domain. And I think when we think of eukarya, we think of ourselves, and we think of our dog, and we think of cows or horses or whatever else that we're sort of used to. So there's multicellular eukaryotic organisms, but then there's also unicellular organisms that fall under eukarya, like stentor and amoeba and other protists. So eukarya is not just multicellular organisms. There's more than several. There's many single-celled organisms in eukarya as well. So eukarya, all of those organisms have a nucleus, and then as far as supergroups goes, Eukaryotic supergroups are kind of currently an area of debate because we're looking at molecular relationships. So there's pictures there from two different textbooks. They don't agree on everything in these two different images. They agree on some things, but all the dotted lines in those graphs are areas that are kind of unresolved. So they're not quite sure the evolutionary history of those different organisms. So I'm not going to make you memorize any eukaryotic supergroups. However, I do want you to know broadly about the eukaryotic kingdoms. So we've got plants, fungi, and animals for eukaryotic kingdoms. We also have a group of eukaryotic organisms that's kind of the catch-all group of eukaryotic critters. So if you're not a plant, you're not a fungus, you're not an animal, but you're eukaryotic, you fall into the group Protista, which again is not truly a kingdom anymore because organisms that are classified as protist are not more closely related to each other than they are other organisms. So if we look at plants, again, plants are eukaryotic. They have cell walls that are made of cellulose. They're photosynthetic for the most part. There's some exceptions of like parasitic plants, but for the most part, they're photosynthetic and they're multicellular. If we look at fungi have cell walls that are made of chitin. They're not photosynthetic and they could either be unicellular or multicellular. Then we've got animals. They've got no cell walls. They're not photosynthetic and they're multicellular. And then we've got that catch-all group, protist. Not a kingdom though, so be careful on that one. Their classification is under construction or under debate. But broadly speaking, we can talk about plant-like protists that are algae. We've got animal-like protists, so we call those protozoans. They're heterotrophic. And then we've got molds, which I put scare quotes around because true molds are fungus, so these are fungus-like protists. So let's classify the pictured organisms. The first one, we've got this purple thing here with green parts on it. That's going to be a plant, so that will be C. This squiggly guy here is an earthworm, so that is A, or an animal. In the middle here, you've probably seen or eaten some of those, or may have seen or eaten some of those. That's a morel mushroom, so that's a fungus. Down here, we've got a fish, so that's an animal, so that's A. And then you're like, what is that thing? It looks like a tornado, or I don't know, a badly made wine glass. Either way, that is a protist. So if we talk about the tree of life and tracing evolutionary history, the current tree of life is based largely on ribosomal RNA genes or rRNA genes. These genes have evolved slowly, so that allows us to detect similarities between distantly related organisms. And other genes sometimes indicate slightly different evolutionary relationships, so we're still working on untangling this tangled tree of life. Horizontal gene transfer also complicates efforts to build a tree of life. And really, horizontal gene transfer is common enough that the early history of life is really better dictated by kind of a tangled web than a truly branching tree. Also, this tree of life suggests that eukaryotic organisms and archaea are more closely related to each other than archaea is to bacteria, which is interesting. So there's kind of two depictions of the tree of life. This first one on the left-hand side is okay, but they've got, multiple branch points coming off of this common ancestor of all life. So there's some unresolved areas there. This next picture where it's like this tangled web is more helpful in some ways because it's showing you horizontal gene transfer, but it's more complicated in others. They've highlighted mitochondria and chloroplast, going from bacteria to, plants for chloroplast, and plants, animals, and fungi for mitochondria. So they're showing horizontal gene transfer there. But as far as that goes, I mean, there's still some things to work out with that as well. So again, horizontal gene transfer has really kind of tangled up that tree of life, and there's evidence for substantial interchanges of genes between organisms in different domains. Horizontal gene transfer occurs by exchange of transposable elements. So those are those jumping genes in DNA. Exchange of plasmids. So remember, plasmids are those extra chromosomal pieces of DNA in bacteria, or a viral infection, or even fusion of organisms, like an organism taking in another organism, like in the case of chloroplast and mitochondria. So as far as naming things go, we name things with a two-name naming system. So binomial nomenclature is two-name naming system. The first name is the genus. The second name is the specific epithet, which is the descriptor. And then when we have the first name and the second name together, you get the species name. So as far as writing this down goes, this is a kind of big deal. I always tell this story, but when I was in herpetology class, which is the study of like reptiles, we had a lab exam, and I spent forever, or it felt like forever, studying these reptiles. And then, you know, we get to the lab, we go around, we identify them, you know, and then our TA goes and grades our exams. I missed every single question, even though I got all the answers right, because I did not correctly write the names. So I didn't underline them. So my TA counted every single one wrong. Right? So basically, I want you to not make that mistake. I want you to go into your herpetology quiz or your woody plants quiz or whatever, and I want you to underline all of your scientific names so you don't miss any points. So another thing you need to do is make sure your capitalization is correct. So you should only capitalize the first letter of the genus, and then both names need to be underlined if it's handwritten, or it needs to be in italics if it's typed. And after you've already used the genus one time, you can abbreviate it, although you might not want to do that on your quiz depending on your TA. But you can abbreviate it after you've already written the genus fully one time. So we use binomial nomenclature to avoid confusion. Common names can vary from one region to another. Also, we use binomial nomenclature to avoid false classification. Like if we look at the koala bear there, koala bears are not a bear. Koalas are marsupials. So it should be like the koala marsupial, but no one calls them that. It's very confusing. Another example, if we look at the picture there of the common sunflower, common to where, right? Common to your region, common to everywhere, who knows? So not a particularly great name. But if we wanted to research that particular sunflower, we could research it under its scientific name. And again, there's 108 types of sunflowers in the United States, so calling it the common sunflower is probably not that helpful. So biologists and scientists always like to name and sort things. So phylogeny and taxonomy are a little bit different. Taxonomy is really naming and classifying, and then phylogeny is trying to trace an evolutionary history of a species or a group of related species. So sometimes the naming and describing doesn't necessarily line up with the tracing the evolutionary history. So the name classification and phylogeny can sometimes differ from one another. And if anybody has ever complained about like, oh, those scientists are always renaming things, which is common when you're like a little bit into science but not maybe super far, or maybe even if you're super far into science, but you get frustrated at having to relearn names of things. We are trying to kind of get them together, but they're not always going to line up. But anyhow, phylogeny can lead to classification. Systematics is going to classify organisms according to evolutionary relationships, and cladistics is looking more at order and time that the branches arise. So these kind of all go together, but they're all a little bit different. On your exam, I'm not going to catch you up and try and trip you up on these terms. So we can depict evolutionary relationships in a phylogenetic tree. A phylogenetic tree is a diagram that illustrates phylogeny, or evolutionary history. It's really a hypothesis of the evolutionary relationships among various species, and we infer these from similarities based on available information. So originally this was done with morphological data, so what things look like, and now it's done with molecular data as well. And the best the best phylogenetic tree fits the most data. So it fits morphological data, molecular data, and fossil evidence. And the cool thing about this is when we get new evidence, we modify the phylogenetic trees. So here's just an example of a phylogenetic tree here. And you can kind of see we've got the wolf and the coyote sharing a common ancestor. And then you've got the canine family sharing a common ancestor with the mustelid family, and kind of so on and so forth there. So when we look at organisms with similar or shared structures, it's important that we understand if the similarity is a result of a homology or an analogy. So homology is when we have similar structures due to shared ancestry, and analogies are when we have similarities due to convergent evolution. So convergent evolution occurs when similar environmental pressures and natural selection produce similar structures or adaptations from organisms that are found in different evolutionary lineages. So we have the same kind of structure in organisms that are not closely related to each other. A homoplasy is an analogous structure or an molecular sequence that evolved independently. So homology can be distinguished from analogy by comparing the fossil evidence and the degree of complexity. So if we look at burrowers, they're going to have some convergent evolution happening there. They tend to have a long body. They tend to have large front paws and small eyes and a thick pad of skin that protects the nose. However, if we look at the Australian marsupial mole and the North American eutherian mole, that means it's a placental mole versus a marsupial or pouched mole. They're going to have similar structures, but that's convergent evolution because they don't share a common ancestor with those structures. So the more complex two similar structures are, the more likely it is that they're homologous. However, if you look at the claws and look at the nose pad on these guys, you can see that they're actually pretty different. So if we look at another example of homology versus analogy, so bat and bird wings are homologous as forelimbs because chordates share a common ancestor with forelimbs that have the same or similar bone structure, but as functional wings are analogous. A bird wing is very different than the structure of a bat wing, even though they're evolved for the same sort of reason, or they have the same benefits. And then if we look at an insect wing is even more different. But a bat, a bird, and an insect might even occupy the same sort of niche or the same sort of space in their environment. When we look at molecular homologies are determined based on the degree of similarity and the nucleotide sequence between different taxa. So the more similar they are, the more likely it is that they are the result of having a common ancestor. So systematicists can actually use computer programs to analyze this. And then the figure here is just showing two DNA sequences that at first glance might not appear that similar, but if the first sequence had a deletion and the second sequence had a triplet inserted, the sequences are actually pretty similar. So molecular homoplasies are analogous sequences, so they're shared bases in a nucleotide sequence that are otherwise very dissimilar. So these are not the result of a common ancestor. So in this case, there's few similarities, and they're not very complex, so it's likely not due to sharing a common ancestor with those particular sequences. So let's review then. Giant anteaters and echidnas, so both of those are pictured below, have long snouts and tongues to feed on ants. If these organisms do not share a common ancestor, which term could be used to describe their snouts? So that would be analogous structures. And they do share a common ancestor at some point, but really we should say do not share a recent common ancestor. And then number 2, which of these structures is a structure that's similar because organisms share a common ancestor with the structure? That's going to be B, homologous structure. So when we look at phylogenetic trees, there's some terminology we can talk about, and then we'll look at an actual tree on the next slide here. But the branch point is going to represent where 2 different taxa diverge from a common ancestor. Sister taxa are groups that share an intermediate common ancestor. A rooted tree is a tree that includes a branch to represent the most recent common ancestor of all the taxa in that tree. The basal taxon is going to be a group that diverges really early in the history of the group and originates near the common ancestor of the group. And then a polytomy is when we have a branch from which more than 2 groups emerge. This means we have an unresolved pattern of divergence, and we probably need more data to resolve that. So here's a picture with all of those labeled. So number 1 here is the branch point. That's the common ancestor for all the different taxa, A through G. Number 2 here is a branch point. So the lineages are going to diverge here as well. 5 is showing us a polytomy. So you can see how we've got 3 things coming off of this one branch point. That probably needs more data to clear that up. Spot G down here is the basal taxon. It diverged really early on. And then like B and C here are sister taxa. So with cladistics is going to classify organisms by order and time that the branches arise. So we're classifying by common descent here. A clade is a group of species that includes an ancestral species and then all of its descendants. Clades can actually be nested within larger clades, but not all groupings of organisms can qualify as a clade. Valid clades are monophyletic, meaning that it consists of the ancestral species and all of its descendants. I'll show you a couple pictures on the next slide. So with taxonomic groupings, we're shooting for a monophyletic group or a clade, which consists of an ancestral species and all of its descendants. So for example, a monophyletic group would be everyone in this picture. So the even-toed ungulates, the hippo, whales, seals, bears, other carnivores. That would be a monophyletic grouping. Or if we did seals, bears, other carnivores, that would be a monophyletic grouping or a clade. Or if we just did seals and bears, that would be a monophyletic grouping or a clade as well. A paraphyletic grouping consists of an ancestral species and some of the descendants, but not all of the descendants. So in this one, they left the whales off. Polyphyletic grouping is when we have various taxa that have different ancestors. So here with the whales and the seals, they didn't get their ancestor, which is all the way down here, and they didn't get the rest of the organisms that would be in that clade. So really polyphyletic groupings is why they are rearranging the eukaryotic kingdoms into supergroups and trying to deal with the mess that is Protists. So go ahead and look at the pictures and determine if the grouping is either monophyletic, polyphyletic, or paraphyletic. So if we look at A has the ancestor and all of its descendants grouped together. So that is going to be a monophyletic grouping or a clade. If we look at B B left off this group G down here, so this is actually going to be paraphyletic. And then polyphyletic here, we've got D included. D should not be included, or we should include E, F, and G. So C is going to be polyphyletic. So to build a phylogenetic tree, we're going to do this together in class as well. But first you're going to need to determine your in-group, which is the group of critters you want to study. They should be pretty similar to each other. And then you want to determine an outgroup or an outlier. That is a group that has diverged before the in-group, and they should be related but not as closely related to each other as the members in the in-group. So they should be pretty different. And then if we have characters that are shared by both the outgroup and the in-group, those would be ancestral characters that predate the divergence of both groups from a common ancestor. So then we're going to choose character states that are present in all of the organisms, including the outgroup. So character state is a morphological or molecular trait that exists in a distinct state with no ambiguity. So you should be able to answer all of these as yes or no type answers. So it either has hair or does not have hair, or it has an A on the 10th base of the first chromosome, or it doesn't have an A on the 10th base of the first chromosome. And again, we'll go over these in class, but this is kind of how you're going to do that. Then you're going to create a chart or a data matrix with your organisms along one side and your character states along the other. It's kind of helpful to have a total column as well. And you're going to complete your chart or data matrix by polarizing your chosen character states. You're going to assign a 0 to an ancestral trait. So that would be the kind of trait that would be in your outgroup, and you'll assign a 1 to any derived character states. You're going to total each row, and the outgroup needs to have the lowest number or all zeros because your outgroup was picked because it's the most ancestral grouping. So here in our example, we've got a lancelet as our outgroup. We've got a lamprey, bass, frog, turtle, and leopard. And our characteristics are a vertebral column, hinged jaws, 4 limbs, an amnion, and hair. So the lancelet doesn't have any of that. The lamprey has a vertebral column, so the lamprey gets a 1 there, and so does everybody other than the lancelet. Then on hinged jaws, the lancelet and the lamprey do not have that, so they get 0s, but everybody else gets a 1. On 4 limbs, the fish does not have 4 limbs, but the frog, the turtle, and the leopard do, so they get 1s. With the amnion, the turtle and the leopard do. Like the leopard has amniotic fluid in its placenta, and the turtle lays amniotic eggs. And then hair, the leopard is the only one with hair, so it's the only one that gets a 1 there. So then we're going to write the organism's names from the lowest total on the left-hand side to the highest total on the right-hand side. So our outgroup is going to be on the left-hand side, and the one with the highest number will be on the right-hand side. Then you're going to complete your cladogram by drawing the lines in the cladogram, again from the bottom to the top. The left side should have a longer line, the right side should have kind of a shorter line, and you give 1 branch per organism. You can also include the character states. So in our example here, the character state of having hair, the leopard has hair, but the turtle, the frog, the fish, the lamprey, the lancelet, none of them have hair. If we look at 4 limbs, this is saying the frog, the turtle, and the leopard have 4 limbs, but the bass, the lamprey, and the lancelet do not. So then these 2 cladograms here are equivalent. Usually when you hand draw your cladogram, you're going to create something like the top one. But down here on the bottom, that's more like what your book is going to have for you. But really they are equivalent. And it's not to say like leopard is first place, turtle is second place, frog is third place, or so on and so forth. We're also not saying a leopard evolved from a turtle. Instead, what we're saying is that the turtle and the leopard share a common ancestor. So when we're picking characteristics that we are going to use in our phylogenetic trees, these similarities need to result from homology, so shared ancestry, rather than convergent evolution in analogous structures. It's hard to know that, especially if you are in your Bio 160 class and you don't know a lot about the organisms. You might not pick homologous structures. This requires some research. But we'll just kind of do the best we can in class, understanding our kind of limited knowledge base. We have to realize that organisms have shared and different characteristics, or shared and derived characteristics, when we compare them to their ancestors. A shared ancestral character is a character that originated in an ancestor of the taxon or group of critters, and a shared derived character is an evolutionary novelty to that particular clade or group of critters. So when we infer evolutionary relationships, it's useful to know in which clade a shared derived character first appeared. So let's review again. For species D and E, having two front flippers, is it a shared derived character or a shared ancestral character? If we look, species B has two front flippers, but species A does not have two front flippers. So for this one, for species D and E, having two front flippers is a shared derived character. So 4 is A. If we look at species D and E again, having two eyes, is it a derived character or an ancestral character? In this case, that is a shared ancestral character, so 5 is B. Number 6, which of the images have clades highlighted? So if we look at image A is good to go. There's the ancestor and then all of its descendants. B, not so much, right? We didn't get the ancestor and all of its descendants. C, not so much there as well. We missed one. And then D is a little bit tricky, but D would actually count as a clade. So for number 7, which of these trees depicts an evolutionary history that's different from the other two? Go ahead and pause it here and hit play when you're ready. So the last picture, or C, is the one that's different from the first two. So if we look at and it gets tricky because the labeling is awful here because they're all labeled with letters. But if we look at the tree C, the third tree, it has organisms C and B grouped together. But in the other trees, it's C and D that are grouped together. So this last tree here, the third set, is the one that is different. But A and B, or the first and second group here, are the same. So phylogenetic trees can show us patterns of descent, but not so much phenotypic similarity. Although phylogenies do provide important information about similar characteristics in closely related species, they also don't generally indicate when a species evolved or how much change occurred in a particular lineage. It should not be assumed that a taxon evolved from the taxon next to it. Like I said, the leopard didn't evolve from the turtle. Then phylogenetic trees based on DNA sequences can be used, though, to infer species identities. So your book has a good example on this. They used a phylogeny to identify the species of whale from which whale meat originated. So here's an example of that. They had a couple unknown DNA samples of whale meat, and they determined the species by creating a phylogenetic tree there. So in some trees, the branch length can reflect the number of genetic changes that have taken place, and in other trees, the branch length can represent chronological time. And sometimes the branch length is arbitrary, so be careful and make sure that you're reading like legends when you're looking at these. So systematicists can never be sure that they found the best tree in a large data set. However, they can use Occam's razor and the principle of maximum parsimony. So they would say that the tree that requires the fewest evolutionary events is the most likely tree. And there's computer programs that we can use to kind of search for the trees that are parsimonious or have the least number of evolutionary events. So a lot of times when students look at this picture, they kind of panic and they're like, oh my gosh, there's a whole lot going on. There's 3 birds, there's all these trees, there's all these dashes. What's happening here? So each dash is representing an evolutionary event or a mutation that would have had to occur to produce these 3 different bird types with these 3 different sequences. So this tree here at the bottom, where there were 6 events that had to occur, is the most likely tree because the other trees took 7 events. But don't worry, I'm not going to give you an example like that. Fortunately, we've got computers to help us with this sort of thing. So then we can use something called molecular clocks to kind of keep track of evolutionary time. If we say that over the course of millions of years, mutations are going to kind of build up in a particular stretch of DNA, we would assume that they would occur at kind of a reliable rate. We can use a molecular clock to kind of keep track of time. So with a molecular clock, if the number of nucleotide substitutions in the related genes is proportional to the time since they last shared a common ancestor, that can give us a good measure of time. However, there's some problems with this because some genes are not as clock-like as others. Mutations that are neutral or have little or no effect on the fitness or reproductive success of organisms should be regular and clock-like. However, mutations that have a significant impact on the fitness or reproductive success of an organism are going to be less clock-like, because if those organisms are not biologically fit, they're not reproducing, and so we wouldn't see that particular mutation in subsequent generations. Molecular clocks can be calibrated by plotting the number of genetic changes against the date of the branch point in a fossil record. And if we are looking at more genes, that gives us a better idea of the date using molecular clock. But molecular clocks don't even run as smoothly as expected, even if the mutations were selectively neutral. There's some irregularities that are the result of natural selection, in which some DNA changes are actually favored over others. And there's going to be a problem when we look at estimates of evolutionary divergences that are older than the fossil record, because we can't calibrate our clock. When my family goes in the store and is trying to find each other later, we'll like pick a meetup point and pick a time that we're going to meet up, and everybody has a phone now, but before you had a phone, you would look at your watch and you would say, let's synchronize our watches. And then, my dad would be like, okay, meet by cash register 3 at 10:15, and we'd look at our watches to make sure that all of our watches were paired up and correct, and then we'd go to the store and then we'd meet back at cash register 3 at 10:15. So if you can't calibrate your clock, it doesn't matter. Like if your clock says it's noon and it's not really noon, that's a problem. So there are some problems with molecular clocks, but if we look at multiple genes, that can improve our estimates. So we talked about taxonomy, which is naming and sorting things. We talked about the 3-domain system. We talked about eukaryotic kingdoms. I do want you to know your domains. I do want you to know your kingdoms. And I do want you to know the hierarchy. So domain, supergroup, kingdom, phylum, class, order, family, genus, species. Make sure you memorize that. We talked about binomial nomenclature, the 2-name naming system. So first name, genus, capitalized. Second name, specific epithet. Together that makes the species name. If you're handwriting it, underline it. If you're typing it, italicize it. We talked about the difference between phylogeny, systematics, and cladistics. Don't get too hung up there. We talked about homologies, so similar structures or sequences that are due to shared ancestry, and analogous structures, which are structures that are due to something like convergent evolution, or they could even just be like a random mutation that occurred in different organisms. So those are structures that are not similar due to common ancestry. And then we talked about building a phylogenetic tree. We'll do some examples of that in class. We talked about clades, which are when we have an ancestral species and all of its descendants. So a monophyletic grouping would be a clade. And then we talked about paraphyletic and polyphyletic groupings, which are really taxonomically or systematically not a good kind of grouping there. And then we talked a little bit about molecular clock and some of the problems with the molecular clock. So that's it for this chapter.